The Meselson-Stahl experiment proved that DNA replicated semi-conservatively. They used E. coli bacteria to prove this replication process. So to do that, first of all, they grew this E. coli with a heavy isotope of nitrogen. They did this continuously for several generations. So the reason they did that was to make sure that all of the E. coli's bases would contain nitrogen 15 instead of nitrogen 14. So essentially you've made DNA molecules that contain these heavier bases. So as I mentioned, the nitrogen 15 isotope gets incorporated into all of the bases, and so after this point in time they all contain this heavy type, nitrogen 15. One of those samples that is grown purely in nitrogen 15 gets saved for later. The E. coli is then transferred to a medium that contains nitrogen 14. This is the normal lighter isotope of nitrogen. So now the new DNA that is synthesized will be synthesized with this isotope as opposed to the nitrogen 15, because there's no other nitrogen that's available to form these bases. The E. coli then get left to grow and divide. Now the reason that you choose bacteria for this, as well as their simplicity, is the fact that they replicate really quickly, so around 50 minutes. After that point, a sample is taken. After the E. coli get divided a second time, another 50 minutes, a second sample is taken. So we've now got the original nitrogen 15, we've got the first sample, and we've now got this second generation sample. The DNA then is extracted from all the three samples and added to a solution of cesium chloride, which helps to separate the DNA out based on density alone. DNA is also taken from a control sample of E. coli, which has been grown exclusively in nitrogen 14, exactly the same as the nitrogen 15 original sample. So what that means is we now end up with a nitrogen 15 only grown sample, nitrogen 15 sample which is a first generation which was combined with the nitrogen 14, a second generation that was grown with the nitrogen 14, and then a control sample that has only nitrogen 14. These tubes then get spun at high speeds in a centrifuge, and each of the tubes the DNA will then collect into a band, so the heavier the DNA the lower down the tube that band will form. If we compare the position of the bands from the different E. coli samples, that will provide evidence for the fact that we've observed a semi-conservative method of replication. If we look at each result individually, the nitrogen 15 will form a band near the bottom of the tube. Now this one has only strands formed from bases of nitrogen 15. If we have a look at the nitrogen 14 control tube next, that is what we would term the lighter DNA. That one forms a band higher up near the top of the tube because that contains only nitrogen 14, so a lighter version of the nitrogen. The first generation will form a band midway between the heavy and the light bands we've just seen. So that intermediate DNA strand will contain one strand that has been formed with nitrogen 15, so the original strand, and then through the process of semi-conservative replication, the second strand is made with nitrogen 14. The second generation will have bands at 2 points. So it will have the ones from the first generation, which have one heavier strand and one lighter strand, and it will also have ones composed of just lighter strands, because there will be molecules which are formed using the original existing strand and the nitrogen 14 strand, which therefore, if it's grown in a nitrogen 14 solution, it will only have the lighter strands as part of the composition of that one. That then gives evidence for the method of semi-conservative replication, because we can see that as we move through the generation, the first one has got this intermediate band because they will all be composed of the nitrogen 15 bases and the nitrogen 14 bases, And then comparing it to the second generation, we can see that we will have ones that are composed of just nitrogen 14 and the nitrogen 15 and nitrogen 14